My name is Ya-Qing Zhang. I'll introduce the paper submitted to iMeta. The title is A Gut Microbiota-Bile Acid Axis Inhibits the Infection of an Emerging Coronavirus by Targeting its Cellular Receptor Aminopeptidase N. In 2006 and 2009, researchers in Hong Kong identified 3 novel coronaviruses in wild Asian leopard cats and bird populations. Genomic analysis in 2012 confirmed these as deltacoronaviruses, CoVs, with PDCoV first detected in pigs. In 2014, PDCoV outbreaks in the United States caused severe diarrhea and vomiting in piglets, leading to global spread and significant economic losses. Recent studies have highlighted PDCoV's expanding host range and its potential for cross-species transmission. Evidence suggests an avian origin, with PDCoV capable of infecting mice, chicks, and even detected in plasma from febrile children in Haiti. Our lab first identified APN as the cellular receptor PDCoV in 2018 and demonstrated in 2021 that the virus exploits APN from humans, mice, cats, and chickens for cell entry, with sialic acid as a co-receptor. In 2023, we reported that 3 avian DCoV pseudoviruses could invade cells via chicken or porcine APN, providing a foundation for understanding PDCoV's cross-species transmission. Current research on PDCoV infection mechanisms focuses narrowly on virus-host interactions. Our study investigates the impact of PDCoV on gut microbiota in piglets, identifies key bacteria, and elucidates the regulatory role of their metabolites in infection, providing new insights for controlling PDCoV or coronaviruses. Key findings: PDCoV-infected 3-day-old piglets exhibited typical diarrhea and ileal damage, villus atrophy, crypt hyperplasia. 16S rRNA and metagenomic analyses revealed significant alterations in gut microbial composition and function. Notably, Escherichia coli, Bacteroides fragilis, and Clostridium perfringens were markedly reduced, showing negative correlations with viral load. B. fragilis abundance declined progressively with infection, strongly correlating with viral replication and pathology. Untargeted and targeted metabolomics showed PDCoV infection enriched primary bile acid pathways. Free bile acids, Ca, DCA, LCA, decreased, while conjugated bile acids, GCA, GLCA, increased, linked to reduced microbial bile salt hydrolase, BSH, activity. In mouse models, B. fragilis colonization restored free bile acids, DCA, LCA, via BSH activity, reducing viral loads. BSH inhibitor GR7 abolished this effect. This highlights the critical role of Bacteroides fragilis in regulating bile acid metabolism and antiviral immunity. Further in vitro experiments revealed that LCA dose-dependently inhibited PDCoV infection in porcine small intestinal epithelial cells and intestinal organoids. Mechanistic studies showed that LCA specifically targeted the viral attachment stage by blocking spike protein-mediated membrane binding, and this effect depended on host APN expression. ELISA directly confirmed that LCA inhibits the binding of the S protein to APN. Molecular docking and dynamic simulations showed that LCA embeds itself at the interface between the receptor binding domain and APN, forming a stable hydrogen bond network and inducing conformational changes in the RBD, thereby antagonizing viral entry. In piglet models, oral administration of LCA significantly reduced intestinal viral load, alleviated diarrhea symptoms, and improved tissue pathology without inducing inflammation, apoptosis, or intestinal barrier dysfunction. These results demonstrate its excellent safety profile in the gut and its potential as a transdermal antiviral therapeutic. In summary, our study is the first to integrate multi-omics analyses, uncovering PDCoV-induced dysbiosis of the gut microbiota-bile acid axis and its pathological consequences. We identified B. fragilis as a key modulator of bile acid metabolism via BSH, inhibiting PDCoV infection, revealed LCA's novel mechanism of blocking spike-APN interaction. These findings provide a robust theoretical and experimental foundation for developing innovative antiviral strategies.